Hi, Dr. Burgess here. This topic is cell biology and I will be going through cells, cell specialization, microscopy, and then on to stem cells, the cell cycle and mitosis, transport across membranes, and then looking at gas exchange. So I've split the video into different parts. At the end of each part, for the subunits, I've included some exam questions with the answers to help you with exam technique. So I hope you find it useful, and there will be more videos coming shortly. Hi everyone, it's Dr. Burgess here, and this is a video on cell biology, AQA GCSE Biology, AQA GCSE Combined Science Biology, and this is designed for higher tier. Okay, so let's first look at eukaryotic cells. So eukaryotic cells include animal and plant cells. So you'll need to know the structures of an animal cell. So as you can see here, we have a nucleus which controls the activities of the cell, the cytoplasm in which most of the chemical reactions take place, a cell membrane which controls the passage of substances into and out of the cell, mitochondria, which is where aerobic respiration takes place, and ribosomes, which are where protein synthesis occurs. In plant cells, all of the same features of animal cells are in plant cells, except for three differences. So those differences are the chloroplasts, which absorb light to make food for photosynthesis, a permanent vacuole, which is filled with cell sap, and a cell wall, which is made of cellulose, which strengthens the cell. In your exam, you'll need to know the differences between a plant and animal cell, and also you might be asked to label a cell as well. So here are the similarities and differences. So they both have a nucleus, they both have cytoplasm, but in an animal cell it doesn't have a chloroplast because they don't photosynthesize, whereas a plant does. Cell membrane, both have cell membranes. Permanent vacuole is only found in a plant. The mitochondria are both animal and plant. Ribosomes, both. And then cell wall, only found in a plant. Prokaryotic cells are slightly different to eukaryotic cells. So a good example is a bacteria. So bacterial cells do not have a true nucleus. So they just have what we call a single strand of DNA that floats in the cytoplasm. They also contain plasmids, which are small rings of DNA. They're also much smaller than eukaryotes as well. So you can see the kind of differences there. They're much simpler in terms of their structure, but they have similar structures to eukaryotic cells, except they do not have a true nucleus and they have small rings of DNA called plasmids. You'll also be expected to learn about specialized cells. So these are cells that are differentiated to become specialized to do a particular function or role. And there are quite a few cells that you need to know about. So I'm going to go through them in turn, but you might need to go through this part of the video again afterwards. So firstly, in plants, we've got what we call palisade cells. So those cells are found at the top of the leaf and they absorb light for photosynthesis. And you'll notice one of the key things that they have is that they have lots of chloroplasts, and they also have a regular shape. The reason they have chloroplasts is to allow them to photosynthesize. Other plant cells you've got, and you'll learn a lot more about these later in the course, is xylem cells and phloem cells. So xylem cells is the movement of water, and these are made of dead cells and they're waterproof, and they go from the root right through the stem to the leaf, and they transport water up the plant. Phloem cells, they move sugars and they move amino acids as well, and they're made of living cells, and they move these things all around the plant, up and down. So they require energy from respiration. So mitochondria are required to move the sugars up and down the plant. Root hair cells. So root hair cells are found in roots, obviously. There are millions of them, and you'll notice by the shape they have a large surface area, okay, and they also have very thin walls as well, and they allow water and mineral ions to be absorbed. But the main key things are large surface area and very thin walls. So in animal cells, you'll need to know about sperm cells. So a sperm cell, as you'll probably know, fertilizes an egg cell, and the tail, it can move due to mitochondria in the middle part of it that releases energy. It also has enzymes in the head which breaks down the egg, surface of the egg, so it can gain entry into the egg or ovum. Another animal cell you'll need to know is about muscle cells. So muscle cells, they contract to move the body. So they have filaments that slide over each other to shorten, to contract things. They need lots and lots of mitochondria, and they're really important. And you need lots of energy from respiration, and you need lots of glucose being delivered to those muscle cells. And the third animal cell you need to know a bit about is nerve cells. Now, nerve cells are very long, they're very thin, they're insulated because they're carrying electrical impulses around the body, and they connect to each other. And again, later in the course, you'll learn a bit more about nerve cells. Just a reminder, and I'm going to put the link into the description of this video. There is a required practical here in terms of observing animal and plant cells, and Dr. Biology will go through that with you. Okay, here's a question checkpoint. So this is a bit different to my other videos. So what I'm going to ask you to do is to have a look at these questions, and I'd like you to pause and actually answer the questions yourself before moving on to look at the answers. So if you're going to do that, pause now. Okay, so let's have a look. So the first question, it says draw one line from each cell structure to the type of cell where the structure is found. So they want you to draw one from cell structure to the type of cell where the structure is found. Okay, so the first answer, so nucleus is a eukaryotic cell. Permanent vacuole is found in the plant cells only. And plasmids are found in prokaryotic cells. And then the second one shows a plant cell, and it says what are the names of structures A, B and C. Now this type of question is a bit confusing because you've got to get all three structures correct to get the answer. Now you'll notice there's a tick box on the right hand side, so you need to put your answer there. You need to put a tick in one of these boxes. So you need to go through structure A. So structure A, I'm going to tell you, is a vacuole, so it can either be that one or that one. Structure B is not a great diagram, but usually the smallest part of a cell is ribosomes, so I'm going to go to ribosomes. So therefore C is a cell wall, so it can only be vacuole, ribosome and cell wall. There we go. Okay, next question. This is about specialized cells. So what I would like you to do again is to, in a minute, is to pause the video and to have a go at answering these questions. So 3, 2, 1, pause. And welcome back. So question 2. So the diagram shows 4 types of cells, so it doesn't tell you what types of cells they are. It says 2 of the cells are plant cells and 2 are animal cells. So then let's go to the first question. So which 2 of the cells are plant cells? So you need to look at the clues. Now the first clue is chloroplast, so definitely D. So D is definitely a plant cell, so yeah, it could be either that one or that one. Now let's have a look. So A and D, well I know A is a sperm cell, so it can't be A and D, but C, it says it's a root hair, so it's a root hair cell, so it would be C and D. Which part is found only in plant cells? So draw a ring around one answer. So we know from looking at the differences between plant cells that it is cell wall. Which cell is adapted for swimming? Well, that's A, which is sperm. And which cell can produce glucose by photosynthesis? Well, that will be D, because it contains chloroplasts. And then cells all use oxygen. For what process do cells use oxygen? Well, they use it for respiration. Part 2 is microscopy, so it's about microscopes and looking at and observing cells with a light microscope. So here we have a light microscope. You might be expected in an exam to be able to label these things. So you've got the eyepiece, you've got the objective lenses, that is not actually shown in this diagram, unfortunately. Objective lenses are the ones that you can rotate and they increase the magnification. You've got the stage, you've got stage clips, you've got the microscope slide on the stage, you've got the light, so the light passes up through the lenses, through the eyepiece into your eye, and then you've got a focusing wheel, so it's either a fine focusing or coarse focusing wheel. So microscopes, they use lenses to magnify or to make bigger and increase the resolution of images. So resolution of a microscope is how well it distinguishes between two points that are close together. So it's the degree of detail. The best thing I can describe is if you look on your mobile phone camera, you'll notice that if you try to look at things at great distances, that it might lose its resolution. It depends how good your camera is. So cameras are good at magnifying, but better cameras are really good at giving good resolution. So microscopes are used to view samples that may not be visible to the naked eye, and they allow us to resolve detail that cannot be seen by the naked eye. So let's have a look at the left hand side. So this is about magnification. So here's an actual size of this blue dot, okay, and you magnify it, and that's called high magnification. So when you magnify things, the image is much bigger than the actual size, okay. Whereas if you have low magnification, the image is still magnified, but it's several times bigger than the actual size. If we look at resolution on the right hand side, so if you can see, if you've got high resolution, the image is clearer and it is more detailed, whereas a low resolution, it's less clear, less detailed, and it tends to be blurry. So you will need to know about two types of microscopes. So on the left we have the light microscope, which you've probably used in the lab. So we know that light passes through the specimen. You can view living cells or organisms, which is an advantage, but they do have generally lower magnification and lower resolution than electron microscopes. Now electron microscopes, they don't use light, they use electrons. So electrons have to pass through a tube. You can see the tube there. It's in a vacuum, and that is why specimens are dead, because there's no oxygen in the vacuum. And they give much higher magnification and much higher resolution than microscopes, light microscopes. You'll need to know about the advantages and disadvantages of both. So an advantage of a light microscope is they can be used to study living cells. They are very cheap and they're portable, but they have lower magnification and lower resolution. Whereas electron microscopes, they have higher magnification, higher resolution. However, you cannot study living cells due to the no oxygen and high pressure in the tube. They're very expensive, they're not portable, and also you need a lot of training and a lot of skill to use one, whereas in a light microscope you can learn the skills very easily. So let's talk a bit about scale and size. So you will need to know a bit about scale and size. So as it says here, the human eye is possible to see a large cell with the naked human eye. So that's what we call 50 to 100 micrometers. The light microscope has a maximum resolution of what we call 200 nanometers, and the max magnification is times 2000. And you can study large parts of cells, organelles, such as the nucleus. The electron microscope has a maximum resolution of 50 picometers, so that's times 10 million. So you can study really small subcellular structures such as ribosomes and plasmid DNA, strands of DNA, chromosomes and things like that. So you will be expected to do some calculations. So what I've got here is what I call the I am triangle. So this is a very good way to remember how to do the different equations. So for example, if you wanted to find the actual size of an image, what you would do is you would measure the image size and divide it by the magnification. Some useful things here, so conversions, so micrometers into millimeters, you can divide by 1,000. If you want to convert millimeters into micrometers, you times it by 1,000. And also you'll need to know a bit about standard form. So what I'm going to do now is just to go through some examples. It's best to use some examples. So here we go with the exam checkpoint. So question checkpoint. So again, what I would like you to do is to pause this and see if you can answer this question. So pause now. So a student used a microscope to observe an animal cell, and it shows the student's drawing, and you can see they've measured it in millimeters, so the length of it, and it says the real length of the cell was 280 micrometers. And it says calculate the magnification of the drawing. So if we go back to this, so magnification is the image size divided by the actual size. So in this case the image size is 112 millimeters and the actual size is 280 micrometers. So what you're going to have to do is convert either the micrometers into millimeters or the millimeters into micrometers. Okay, and what you do is this. So the 112, you can convert it into micrometers, so times it by 1,000, or you could take the micrometers of the real length and divide it by 1,000 to get it into millimeters. So you have to make sure you've got the correct units first. I probably would convert it all into micrometers. That's what I always do with my class. So I always say if you measure in millimeters, to get micrometers, you times it by 1,000. Okay, so if you do that, so let's look at the 112,000 divided by 280. So that gives us times 400. So that would be the correct answer. Let's have a look at another one. Okay, so again, the same as before, I'd like you to have a really good look at this. I'd like you to pause and have a go at this question yourself. So 3, 2, 1, pause. Right, so the image shows some muscle cells from the wall of the stomach as seen through a light microscope. And it gives you what we call a scale bar. So that scale bar is 0.1 millimeters. And it says the scale bar represents 0.1 millimeters. Use a ruler to measure the length of the scale bar. So we'll measure the length of the scale bar with our ruler and calculate the magnification. So what you've got to do is basically measure that distance. So that distance, I measured it as 40 millimeters. So what you need to do then is convert it. Well, you don't need to convert it actually. So you can then just divide 40 with 0.1 and that will give you times 400. So you don't even have to measure the cell. Part 3 is stem cells. So stem cells are undifferentiated cells that can then differentiate it into different types of specialized cells. So there are 2 types of stem cells you need to know about in humans or animals, and that is embryonic stem cells. So the embryo is when the sperm has fertilized the egg and the egg then starts to divide. So they are undifferentiated cells. They have the potential to turn into any kind of cell. The adult stem cells, they are found in places like your bone marrow, so they can only turn into some types of cells such as blood cells. Now you do have other adult stem cells around your body, but they're very, very limited to what type of cells they can produce. So here's an embryonic stem cell, and you can see it has an inner cell mass, and then the cells will eventually, those undifferentiated cells, they will differentiate into any type of specialized cell. So in this case, fat cells, nerve cells, red blood cells, white blood cells, lipids, anything. So they can produce any type of specialized cell. Whereas an adult stem cell, so for example, blood stem cells are found within the bone marrow of your bones, and they can differentiate into different types of blood cells. So they're specifically making different types of blood cell, in this case red, white blood cells and platelets. Now, why are stem cells so useful for us in the future and at the moment? So you could do something called therapeutic cloning. So this diagram is showing therapeutic cloning with embryos. Now, embryos can be harvested from unwanted embryos. So for example, after fertility treatments, some people might have spare embryos that they don't want anymore and they're going to be thrown away. So they can then culture these to produce more stem cells. Chemicals are used to make them turn into different types of cell, and this is obviously at the research stage where you can produce muscle cells, nerve cells, possibly insulin producing cells to cure people with diabetes. So they could treat many different conditions and diseases. But the key ethical question we need to think about is it right to destroy an embryo to get to the stem cells? So scientists have thought about this, and they've discovered a new way of using adult cells and adult stem cells and making them behave in a way like an embryonic stem cell. So what you can see here, you can see a human egg cell. So it's not an embryo, it's a human egg cell. And what they do is they remove the nucleus, and then they take the nucleus from a patient cell and they transfer it into the egg cell, and they stimulate the cell to divide, and then the cell develops itself into an embryo. And then after 4 or 5 days you can remove those stem cells, they culture them, that means they grow lots of them, and then they could be injected back into the adult patient to try and cure a specific condition. Obviously this is all research at this stage, but there's a real advantage here, and the big advantage is that you're using an adult stem cell, you're not destroying an embryo, so people can give their consent and say that they are happy for their adult stem cells to be used. In exams, they like to give you lots of information about stem cells, and there are different arguments for and against doing stem cell research, particularly with embryos. So these are a couple of arguments for and against. Obviously, I think there are more arguments, and we'll look at that when we go through an exam question. So for stem cell research, it's curing patients. You're curing patients with stem cells. Is that more important than the rights of embryos? So actually trying to relieve suffering of people who are living. So they're just using unwanted embryos as well that might be from fertility clinics that would normally be destroyed. Those against stem cell research would argue that embryos are human life, so you're destroying a life. Scientists could also find other sources of stem cells, for example using adult cells in therapeutic cell cloning. So stem cells are also found in plants. So they are found in what we call the meristem. Now there's a lot of information here. You don't need to know all of this information. All you need to know is that the meristem is basically the areas of growth in the plant. So wherever there is growth, so the top of the stem, for example, to produce new leaves, or in the stem itself, or actual growth within the roots to make the roots longer. So they're able to produce clones of the plant. They can be used to grow crops with specific features for a farmer. So for example, disease resistance. So you could then develop a plant with disease resistance, then clone that plant through taking cuttings or doing something called tissue culture. Okay, so we're on to the question checkpoint. Again, as normal, what I would like you to do is to have a good read of these, and I'd like you to pause and have a go at answering them. So 3, 2, 1, pause. Okay, so let's look at the questions. So first one is quite simple. So stem cells are undifferentiated. Which statement about stem cells is correct? And you literally need to go through the list, and you'll find that actually meristem cells in plants can differentiate throughout the life of the plant. It's a case of going through each one and seeing which ones are completely incorrect, and therefore you can find the correct one. Okay, so this one here is more kind of what you'd expect to see in a higher tier paper. So they give you lots of information about stem cells, therapeutic cell cloning, and they're talking about advantages and disadvantages. So I'm going to show you the answers for advantages. Now, you only need 2 of these answers, okay, but you could write these down maybe with a green pen. So advantages are that you could cure or treat disease, so you don't need to have in your answer both cure and treat disease, just one of those would be fine. It also replaces cells, tissues and organs. Cells are unlikely to be rejected because they're your own cells, so your white blood cells are not going to attack the new cells because they are your cells. And obviously, if you're waiting for a transplant, if they could grow spare organs, then you would not have to wait for a transplant as long. Disadvantages, obviously, with embryos, if you use embryos, life is killed or destroyed. There might be a shortage of eggs or donors. The key thing is we don't know what the risks and side effects might be. Okay, and also a key thing here is if the cell contained a viral infection, you actually can transfer that viral infection into those stem cells. So you could actually transfer viral infections, which is quite severe. It could cause you lots of problems. Part 4, the cell cycle. So before we talk about the cell cycle, we just need to talk a bit about the nucleus, what's within the nucleus, and talk about chromosomes and DNA. You will be learning a bit more about this later in the course as well. So in the nucleus of a human cell, there are 23 pairs of chromosomes. So they contain a double helix of DNA. The chromosomes, they have a large number of genes. In fact, you have about 30,000 genes in your chromosomes. So each section of a chromosome that contains the code for the production of a particular protein is called a gene. So genes code for proteins, and then proteins are made from amino acids, and they make structures such as enzymes. So it's really important to replace these cells on a regular basis. So the cell cycle, very simply, is the way that your cells divide and produce copies. So the first stage is cell growth. So that's when DNA is replicated, or it's a copy is produced of that DNA, and they grow, and then mitosis occurs. Now, mitosis is a form of cell division where you're producing genetically identical cells. So as you can see from this diagram, you can see the cell begins to divide, and the DNA replicates to form 2 copies of each chromosomes, and then the nuclear membrane breaks down and the chromosomes line up in the center of the cell, and then one set of chromosomes is pulled to each end of the cell, and then the nucleus divides and the cytoplasm separates and it divides to form 2 identical cells. When it divides, it's known as cytokinesis. So let's have a look at some questions on this. Okay, so first of all, I'd like you to pause, have a read, pause, and have a go at the question. So 3, 2, 1. Okay, so this one shows a picture of cells dividing in the cell cycle. and it says stage 2 is mitosis, and it shows a simplified cell cycle for a human body cell. And it says draw one line from each stage to what happens during that stage. So stage 1 is going to be copies of DNA are made, so that's in the first stage. Stage 2, the nucleus starts to divide, and stage 3, the cells divide into 2. Here's a harder question, okay? So it says describe what happens in each of the 3 stages of the cell cycle. So using this video and probably your revision notes, I'd like you to pause and have a go at this question. I'd like you to spend about 5 minutes doing this, okay? Off you go. So let's have a look at the answers. So first of all, stage 1, so it does say the 3 stages. So stage 1, you can either have the cell is growing, or there's an increased number of organelles, or you can say there's an increased number of ribosomes or mitochondria. The DNA then replicates, and that's really important that you say that it replicates. Stage 2 is mitosis, so one set of chromosomes moves to each side of the cell, the nucleus divides, and then stage 3, cytoplasm divides to form 2 identical cells, or you could quite simply just say cytokinesis occurs. So you get 4 marks if there's a correct sequence, okay? You've got to mention the stage numbers though in your answers to get the full marks. Part 5, transport across membranes. So this covers quite a large section of work, but I would say this section really does cut across the whole of your GCSE. So I'm going to spend a tiny bit more time on these 3 things because I think they're very important. The 3 areas of transport across membranes is diffusion, osmosis and active transport. And then what we need to do is apply that information into some real world examples within animals and plants, and we'll look at how these organisms allow gases to exchange, to water to move between membranes, and for gases and solutions mixtures to be able to be moved against concentration gradient. So there's lots of information here, but as I said to you, it's really popular in the exams, and they like this this section of work, and they use it a lot in different topics. So let's get started. We'll start off with diffusion. Now I'm going to ask you to learn a set definition. So this definition here, diffusion is the spreading out of the particles of any substance in solution or particles of a gas, resulting in a net movement from an area of higher concentration to an area of lower concentration. So what we have here is a cell, and you can see inside the cell there are some, let's say that's oxygen, okay, and there's less oxygen inside the cell, and there's a higher concentration outside the cell, and what will happen is that there'll be a net movement from outside to inside the cell. So particles move from a higher to a lower concentration. Diffusion is affected by different factors, so different factors affect the speed of diffusion. So things like temperature, concentration gradient and surface area of membrane. So this will be quite important when we talk about gas exchange in a bit. So first of all, temperature. So the higher the temperature, the faster the rate of diffusion. So as you can see by this picture, that in hot water, this blue ink spreads more quickly from a higher to a lower concentration until an equilibrium is reached. Concentration gradient. It's quite difficult to understand what a concentration gradient is, but I like to think of it as a slide. So here we've got A and B. So A is a steep slide, B is a more shallow slide with a shallow gradient. Okay, and what happens is the steeper the concentration gradient, the faster diffusion takes place. So as you can see, if you've got a higher concentration and a very low concentration, there's a fast rate of diffusion. Whereas if you have a less steep concentration gradient, where you've got concentration gradients that are similar, there's a slower rate of diffusion. Still going from a higher to a lower concentration, but it is slower. But this is the key thing you need to remember. The steeper the concentration gradient, the faster the rate of diffusion. Surface area of membrane is really important. So for example, here I've got a fish gill. So fish gills have large number of gill filaments. These gill filaments are tiny, they're microscopic, and they increase the surface area of the gill. Another example is our lungs. So we're going to talk about our lungs in a minute. In our lungs we have millions and millions of alveoli, and those increase the surface area. So basically, the larger the surface area, the faster the rate of diffusion. So on to the third and final transport across membranes process, which is active transport. So active transport moves substances from a dilute solution to a more concentrated solution. So that's against a concentration gradient. So this requires energy from respiration. Okay, so you can see in this diagram, you've got the cell membrane and you've got a dilute solution here. So it's a low concentration of particles, and they've got to move against the concentration gradient. So there's a more concentrated solution on this side of the cell. So you need energy from respiration in the mitochondria to, energy is required to move it against that concentration gradient. Quick recap on respiration. So you should know that respiration does release energy. So energy is needed to move molecules against a concentration gradient. And aerobic respiration takes place in the mitochondria of the cell. So you can see the word equation for aerobic respiration. So here's a diagram to try and explain it to you. So if you think about diffusion and osmosis, you've got a higher concentration, and basically it's a bit like passing a boulder down a hill. So it will go down really easily. It's known as passive transport because no extra energy is required. Whereas we look on the right hand side, and if you imagine you're trying to push a boulder up a hill, you're going to need to use a lot of energy. So when you move from a lower concentration to a higher concentration, then it will move sorry, that's my dog. So move from a lower to a higher concentration. So this does require energy. Okay, particles move against concentration gradient, and the energy is needed to move molecules from a low to a high concentration. I'm now going to go on to part 6. So I'm not going to go through some questions, because the types of questions that you get for this section of work on transport across membranes are very applied. So they give you specific examples. So I'm going to do that at the end of part 6. So let's start part 6. So part 6, gas exchange. There are 3 main things that you need to remember. So I'm going to give you some specific examples that are mentioned in the specification, but these 3 things are all you need to know about gas exchange when you look at some information that they might give you. And the key things are: to increase gas exchange, you need a large surface area to increase the rate of diffusion. You need thin membranes to provide a short diffusion pathway, and you need to maintain a steep concentration gradient. So for example, blood moves substances quickly, or the lungs move gases quickly. So in the exam there will be there could be 3 different examples, 4 different examples, sorry. So in humans it's the lungs, the villi, and then in plants you're going to look at the leaf, and then in fish you need to look at fish gills. So as it says here, multicellular organisms have a large surface area to volume ratio. So in lungs you have many millions of alveoli. They've got a massive surface area, they're thin, and they've got a really good blood supply. So you can see the blood moving past the alveoli. So when you breathe in, the oxygen is in a much higher concentration inside the alveoli than it is in the blood. So that means that oxygen will diffuse very quickly into the red blood cells and then be moved away. And you can see the deoxygenated blood coming here, and carbon dioxide in the plasma is in much higher concentration. So when you breathe out, it moves across the membranes, through the alveoli, and then out of the alveoli in your lungs and through your mouth. The other example is the villi. So in your small intestine, you've got millions of villi that line the small intestine, increasing the surface area, and they are a single layer of cells, and they've got a really good blood supply, so they're maintaining that concentration gradient. Exchange in plants is slightly different. So here we've got a leaf. So this is the top of the leaf, and you can see the palisade cells, the spongy mesophyll, the stomata, and the stomata is where oxygen and carbon dioxide will diffuse in and out of. So the surface of the leaf is very thin and it's flattened, so that increases the surface area for more gas exchange. So in the spongy mesophyll, you've got air spaces that increase the rate of diffusion, and they diffuse in and out of the stomata. Guard cells open and close the stomata, controlling water loss. And then exchange in fish. So fish have gills, and they have many gill filaments, as this diagram shows. And you can see that water enters the fish through the mouth and it goes out through the gills. The oxygen is transported from the water to the blood by diffusion. Each gill has gill filaments, which give the gills a large surface area, and it says lamellae. You probably don't need to know this, but they cover each gill filament to further increase the surface area. They've got good blood supply. They're very thin as well. So this is all about maintaining concentration gradients because of the movement of water and also the movement of blood. You've got thin walls and you've got a massive surface area. Okay, so let's look at some questions related to transport across membranes, including diffusion, osmosis, active transport, and gas exchange. So as usual, I'd like you in a minute to pause this video, and I'd like you to really have a go at doing this question. So pause in 3, 2, 1. And welcome back. So the diagram shows 4 ways in which molecules may move into and out of cells. So the dots are showing the concentration of molecules. So they've got the cell is respiring aerobically, so that means with oxygen, and it says which arrow, A, B, C or D, represents the movement of oxygen molecules. Well, we've got to remember about diffusion. It moves from a higher concentration to a lower concentration, so you need to look at the diagram to work out, well, which has a higher concentration. Okay, and basically the and therefore moving in, and that will be A. So question is A. So movement of carbon dioxide molecules, well, that would be B, because there's more inside the cell and there's less outside the cell. So movement of carbon dioxide out of the cell. Name the process by which gases move into and out. It's diffusion. You can't have osmosis because it's related to gases of oxygen and carbon dioxide. C. Which arrow represents the active uptake of sugar molecules? Well, that would be from a lower to a higher concentration. So in this case, it would be C, because it's uptake, and the reason being because there's uptake against a concentration gradient. Okay, so that's the idea, that it's against the concentration gradient. There we go. So there are the answers. Okay, question 2. So this is about gas exchange. So this shows part of the lungs. So again, what I would like you to do is spend about 3-4 minutes answering this. So please do pause in 3, 2, 1. Okay, welcome back. Right, so give 2 ways the lungs are adapted for efficient exchange. Describe how each adaptation helps to maintain efficient gas exchange. So they want you 2 adaptations, so 2 features or structures, and then a description of each. So you can have many answers here. So for example, adaptation 1, there are lots of alveoli. Okay, and then to get the second mark, you need to say, why are there many alveoli? Well, that means that it provides a larger surface area. You could have good blood supply because they show lots of blood vessels. And what's that doing? Well, that maintains a steep diffusion gradient or concentration gradient between the blood and the alveoli. It's also got very thin walls. You can't really see that by that diagram, but they do have thin walls, so it basically means that diffusion rates are quick because it doesn't have a large distance to travel. You could also have about ventilation as well, so when you breathe in and out, you're maintaining a steep diffusion gradient. So there are 4 examples. You only need 2 of those examples, but it's important you write down the adaptations. So in other words, the structure or the fact that it's a thin wall or good blood supply, many alveoli or well ventilated, and then the description. Okay, so that brings us to the end of cell biology. I hope you found it useful. There is a lot of information there, but I have been as comprehensive as I possibly could. The only thing I would say is that you really do need to go away and look at the required practicals for this unit of work. I will put in the description where you can find those. So if you haven't already subscribed, please do subscribe. And I hope you find it useful, and there'll be more videos like this coming in the future, so I will see you soon.